first speaker is no other than Dr. Dakota Rowsey. He is the Vertebrate Collections Manager at the Arizona State University in Tempe, Arizona, USA, and his talk is entitled Baletemys Kampalili, a new genus and species of shrew-like mouse from a new center of endemism in eastern Mindanao. A brief background of Dr. Rowsey. He oversees the care and maintenance of natural history specimens that comprise of fishes, amphibians, reptiles, birds, and mammals. Also, he works to ensure that these specimens and their associated data are maintained to the highest standard of quality and accessibility for teaching, research, and outreach purposes. With his research, he enjoys questions that use multiple aspects of museum specimens, including skeletal morphology, DNA sequence information, parasite diversity, and more. Dr. Rowsey, you may have the floor.
Thank you so much. Let me start by sharing my screen. Okay. Thank you, everyone. It's so great to be here. I'm truly honored to be invited to the celebration of the UPLB Museum of Natural History, especially because the UPLB is the alma mater of Danny Balete, for whom we named this species. So I'm going to be talking about a recent description of a new genus and species of shrew-like mouse discovered in eastern Mindanao, and the description of a new center of endemism based on this research. This is an international collaboration with individuals from various institutions across the United States and the Philippines, and it's great to be able to represent this this collaboration. So, as an oceanic archipelago, the Philippines is rich with endemic species. In some cases, per unit area, richer than any other place on Earth. For example, on Luzon Island here, 95% of the native terrestrial small mammal species, so rodents and shrews, are endemic to just this single island. And for the most part, these patterns are true across the archipelago and across different vertebrate groups. So whether you're examining birds, frogs, or mammals, the pattern is one of the Philippines being a hotspot for endemic species. And that is illustrated in part by this figure. So just looking at 4 islands in the northern Philippines, Luzon, Lubang, Mindoro, and Sibuyan, with respect to native terrestrial small mammals, for instance, Luzon has 51 species of native terrestrial rodents and shrews, of which 48% are found only on that island. With Lubang, it's 4 and 3, Mindoro, 14 and 11, and Sibuyan, 5 and 4. So this is a pattern that holds, as far as we know, across many different regions in the Philippines. So why is this the case? The Philippine archipelago consists of a few large old islands, such as Luzon and Mindanao Islands, that have persisted in some form for, in some cases, throughout the past 25 million years. This is very old for an oceanic island. A popular example for comparison is the Hawaiian archipelago, which range in the area of 5 to 10 million years at maximum. So throughout the past 25 million years, with again the example of Luzon Island, we see a pattern of originally these islands occurring as single montane regions with some active volcanism, and then as time progresses, we see some additional uplift of other regions, but persistence in this case for the landmass that would eventually become the Central Cordillera of Luzon Island, but then particularly within the last 5 million years of a period of rapid volcanism, where we see uplift of several different regions that would eventually coalesce to become a single island with several different highland montane regions. This is best exemplified with Luzon Island, but we see the same sort of pattern occur even in relatively younger islands, such as Mindanao Island here, parts of the Visayas, and so on and so forth. And so it has been that when researchers have traveled to montane regions across the archipelago, that they have usually found that these regions, these montane regions, have species richness that is much higher than what is previously known, with entire communities of mammals being endemic to a single mountain or mountain range. And that is exemplified by this series of figures here, describing the pattern of species discovery that has followed field expeditions to various montane regions across Luzon, such that in the early 1900s up through almost the 21st century, much of what was known about the endemic small mammal diversity consisted of mostly the Central Cordillera with some representation from the Sierra Madre. But over the past couple decades, field expeditions to various regions throughout Luzon Island have uncovered various communities of endemic small mammals found in some cases on a single mountain and nowhere else in the world. So Luzon is the best represented example of this with small mammals, although some work has begun to be published illustrating the results of field expeditions to other other islands and island groups, such as Mindoro, Lubang, and Sibuyan. This work is still ongoing, and as I'm sure you know, most of the islands across the Philippines have rugged montane regions where we might anticipate additional undiscovered species richness. Such was the thinking behind two separate expeditions, one in 2007 and one in 2010, to Mount Kampalili in eastern Mindanao, represented here with this asterisk. Mindanao Island itself is quite a bit younger than Luzon Island. Our best estimates in terms of its geologic age are perhaps 15 million years or so old. But that is still potentially plenty old enough and large enough as the second largest island in the Philippines to have its own suite of endemic small mammal communities. And so this figure, in addition to illustrating the locations of these field expeditions, also illustrate potential other areas with endemic mammal assemblages based on the fact that they have regions above 1,000, in some cases above 2,000 meters elevation. So these two expeditions to which I refer were planned and led by Danny Balete, a UPLB alum, in collaboration with the Philippine Eagle Foundation. These expeditions yield several unknown organisms, previously unknown organisms, which were found in the high elevation montane forests of Mount Kampalili. For example, one of the new species that was discovered from this expedition were the first small flowered Rafflesia species from Mindanao Island, which is also the only named Rafflesia to occur in montane forests in the Philippines. However, in addition to this new Rafflesia species, one of the major discoveries of this trip was a strange-looking mouse that had a lot of shrew-like qualities. And just for the non-mammalogists in the webinar, shrews and rodents are very distantly related to one another, with shrews being more closely related to cats and dogs than they are to rodents. So these rodents were collected on both the 2007 trip and the 2010 trip, with a total of 3 individuals collected between 1,640 meters elevation and 1,900 meters elevation on the mountain. So well up into the montane forests of of Mount Kampalili. So when I say that this new, previously unknown mouse had a lot of shrew-like characters, I'm referring to it has things like small body size relative to other rodents. It has a slender, elongated snout with narrow incisors, small eyes, short ears, a stout, robust body with a short, tapering, hairy tail, short and very dense fur that is dark both dorsally and ventrally on top and bottom. And it had robust forepaws with relatively long claws, suggesting that it did some level of digging. Now, all of these years later, we were interested in determining what this rodent is related to and how it differs from the other rodent species, both in terms of its morphology, its appearance, as well as its molecular information, its DNA. So we used a mixture of DNA sequencing and skull measurements to determine how different this new this unknown animal was from species that we already knew about, and to determine how different it was. In terms of looking at its DNA, we used gene sequences from 7 loci for 53 different rodent species, and constructed phylogenetic trees to measure the similarity between this new animal relative to other members of the family to which it belongs. In addition, we were interested in estimating how long it has been since this new species diverged from these other rodent species, both within the Philippines and outside of the Philippines. We also collected 26 measurements, 8 from the body and 18 from the skull of this new animal, as well as 38 other individuals to determine how this new species differed from its close relatives. So what I'm showing you here is the results of the phylogenetic analysis for the DNA sequence data of the new species and various other rodents found both in the Philippines and outside. It's, relatively close relatives. So this molecular phylogeny has a scale of time in millions of years ago from the present on this x-axis here. Individuals that are found or endemic to the Philippines are in these gray boxes, and I have the tribe names represented with these bars here. So what you can see is this new animal is a member of a clade containing a bunch of other species that are primarily found and restricted to, endemic to Mindanao Island, with the exception of just a couple species here. Rattus everetti occurs on Mindanao, but is found throughout much of the oceanic Philippines. And there's another species of rat-like rodent that is endemic to Sibuyan that appears to be related to this new species. What is also interesting is that its closest relative outside of the Philippines is this species, Tarsomys sodii, which is a very poorly known rodent species endemic to the Indonesian island of Java. There's clearly more work that needs to be done to determine the diversity of these rodents outside of the Philippines and how that relates to the species that are found in the Philippines. Also, from this phylogeny, you can see that much of the diversification within this group happened within the past 2 million years or so. So a relatively recent series of diversification events in contrast to some of the older Philippine endemic clades. Analysis of the morphometric data, so the skull and the skull measurements, shows that this species differs in concrete ways to other members of the clade of rodents that is found in the Philippines. So what you can see here are 2 principal component analysis plots. A PCA, or principal component analysis, basically takes all of your measurement data and rotates that cloud of points. If you have 18 different measurements, as we did, it rotates those 18 measurements to illustrate which components of your data set explain the greatest amount of variation in your data set. So this first axis, as might be expected for data of this type, illustrates that the largest contributing factor to the data set is indicated by body size. So basically, individuals that score highly are large, individuals that score low are small. So the new species, the two individuals we measured, are relatively small, particularly in comparison to Rattus everetti, which is a big rat-like rat. The second component axis is a composite of information about the width of the palate and the narrowness of the incisors. And this series of measurements, while it does explain about 2% of the variation in our data set, doesn't really do a whole lot to illustrate differences between Baletemys and the other species that we measured. However, if we look at the third principal component axis, this tells us information about the length of the incisive foramen, the width of the first molar, and the length of the molar rows. And Baletemys scores almost at the very bottom on this component axis, indicating that it has a large molar row, or excuse me, a large incisive foramen, a broad first molar, and a long molar row. So that sort of provides qualitative or quantitative information behind how this species differs from its close relatives. And based on the phylogenetic trees we constructed and the morphological analyses, we can confidently conclude that this rodent is not only just a distinct species from the other members in its clade, but is sufficiently distinct from its close relatives in terms of its DNA and its morphology to warrant recognition at the genus or generic level. So this rodent species is intermediate in size between, on the one hand, Rattus everetti, which is very large, and it's worth mentioning that these are all to scale. These skulls are all to scale with one another, is very large compared to the new species. It is smaller than both Rattus everetti and Tarsomys apoensis, but it is larger than Limnomys, the two species of Limnomys, which are cute, sort of arboreal, tree-dwelling mice. It looks the most similar to Tarsomys, particularly Tarsomys apoensis, but differs in several respects. For example, it has an even longer, more tapering snout than Tarsomys. It has a thinner and more slender mandible or lower jaw. It has a smaller and narrower incisive foramen. And that's talking about this little gap here. You can see that this foramen is almost half the size of Tarsomys. In terms of its external characteristics, it has smaller ears, a shorter tail, softer, denser fur with fewer guard hairs, and additionally fewer mammae or nipples. Baletemys has 2 pairs, Tarsomys and Limnomys have 3 pairs, and Rattus everetti has 4 pairs. So there are lots of other traits too, and in the manuscript where we describe this new species, like we go on for pages and pages in excruciating detail and lay out all of the differences between this new species and its close relatives. But these are just some of the biggest ones that I wanted to highlight. We chose to name this species Baletemys Kampalili in honor of Danny Balete and in honor of the place in which this new species was first collected. It is important to us to recognize the people and the places that make biodiversity discovery possible. Danny Balete conducted fieldwork amounting to the area of 50 solid months of fieldwork in his career and contributed to the discovery of 50 species. He also contributed several studies of population biology and was altogether a fantastic advocate for Philippine biodiversity and an amazing scientist in general. So we're delighted to name this species in honor of him. So beyond just knowing about the existence of a new rat species, which, you know, I might be biased, but I think that's pretty cool. But what does Baletemys tell us about Philippine biodiversity? Well, we have already known about 2 old endemic radiations that are found in the Philippines. Those are the tribes Phliomyini, or the cloud rats, and Crotomyini, or the earthworm mice. These two clades are primarily most diverse on Luzon Island and have existed in some cases on the Philippines for 10+ million years, if not older. This study helps to illustrate that there is a third, much younger radiation that has nevertheless diversified both in terms of its species richness and in terms of their ecology. So again, Rattus everetti being a large, sort of what you think of as a rat, Limnomys being an arboreal tree-dwelling mouse, and then Baletemys being predominantly, it seems, terrestrial, and perhaps a species that subsists primarily on insects and or earthworms. Additionally, this new radiation that we've discovered of rodents is endemic primarily, or at least their diversity, is centered on Mindanao Island. So it suggests that there's been this third, much younger radiation of endemic rodents in the Philippines. However, there's still much more work to be done to discover the true diversity of this clade and to discover the patterns of how this clade emerged, diversified, and in the case of Rattus everetti, spread throughout the Philippines. Nevertheless, it suggests that it is possible for mammal radiations to occur at much shorter timescales than was recovered with Crotomyini and Phleomyini. So with the results of this expedition, Danny Balete and the other folks who participated in this expedition basically mirrored what was also discovered on Mount Hamigitan, just a little bit south in eastern Mindanao, where there was a whole different suite of endemic species discovered from there. There's ongoing work in other groups, both within and outside of mammals, that suggests that there's undescribed species diversity, suggesting that Mount Kampalili is an important center of endemism for Philippine biodiversity. It's also an important habitat for Philippine eagles. During his 2010 expedition, Danny remarked that there were many areas of the forest, particularly at higher elevation, that were still intact, but much of the lowland forest had suffered considerable degradation. And I'm just going to pull up an article from the Philippine Center of Investigative Journalism that sampled or that used forest covered data from 2003 to 2015, so a little bit dated, but nevertheless indicated that the Davao de Oro province, here listed as its former name, has undergone considerable deforestation, much of which is illegal. So there is some threat to the biodiversity of these areas. Again, this area is important habitat for Philippine eagles, as well as undescribed species of gymnures, such as a relative of this Podogymnura areospinula. This species is endemic to Dinagat Island. There appears to be another species that occurs on Mount Kampalili. There's ongoing work with this animal and other rodents. It's also an important watershed for the region. So the Philippine Eagle Project has been an important collaborator of this work to help us understand Philippine biodiversity in this region and work to protect habitats to enable the native species here, including Philippine eagles, to flourish. We hope that our work promotes cooperation between policymakers and the local people of these areas to protect this important and unique fauna, including its rodents. With that, I just want to take a few minutes to thank all of the people who have made this work possible. It's definitely been a team effort. This includes the staff of the Philippine Eagle Foundation, who have been important collaborators and contributors to this project. All of the folks who participated in the field work, not only collecting the specimens that we use to perform this study, but lugging the equipment, traversing the rugged terrain. It's not easy work. I'd also like to thank the local residents of the areas in which this work was conducted for providing hospitality to the field teams, the Philippine Department of Environment and Natural Resources for providing the permits that allowed the field teams to collect these specimens. As far as the analyses that went into this project, I'm very grateful for the assistance with staff of the Field Museum who provided me with assistance in borrowing specimens and logistics associated with conducting the DNA sequence work. And with that, I would like to, last but not least, thank you all very much for the invitation. It's so great to be a part of this celebration. If there's time, I'm happy to take questions now. If the organizers would rather wait to the open forum at the end, I'm also happy to wait then. Thank you so much.
All right, thank you very much, Dr. Rowsey. And now we move on to a short open forum moderated by Professor Annalie Adsal. If there are any questions, we will entertain 1 to 2 questions. Then if you have any questions for later, well, again, we remind you there is a short panel discussion later on. So, Prof. Annalie,
okay, good morning. Yes, that was very impressive. It's just like walking us through the field, yes, from field to paper. That's the article. Now, we have one question here, Dr. Rowsey, from Dr. Chun Lee. Were you able to collect the parasites on those species?
So I believe that there were efforts to collect ectoparasites when they were located. I don't believe that that the specimens of the new species had any discovered ectoparasites on them. That's the best of my knowledge.
Thank you. Another question? Okay. Well, it seems they are reserving the questions in the panel discussion. Anyway, Dr. Rowsey, I'd like to present this certificate of appreciation. Let me read it. University of the Philippines, Los Baños, and the UPLB Museum of Natural History awards the certificate of appreciation to Dr. Dakota Rowsey for serving as resource person during the UPLB Museum of Natural History special webinar on research and collection, given this 29th day of September 2022 during the museum's 46th anniversary celebration with the theme Continuing Excellence Through Adversity, Creating Partnerships, Fruitful Research, and Digitized Collections. Signed, Marian P. De Leon, Director, MNH. Thank you, Dr. Rowsey.
Thank you very much.